Hi, my name is Grace and this is Sarah. Our research this summer was focused on finding potential taste receptors in fruit flies.
Fruit flies are a great model organism because they're small, easy to maintain, and live a short duration, which also means they reproduce quickly, which aids in creating genetic mutations. Flies have been used for decades, so their genetic tools are well understood. And although flies are not mammals, they still have many genes with homologs in humans. Their anatomy and nervous systems are also related to ours. Many important diseases have been studied using fruit flies, including Huntington's disease and ALS.
Because we are trying to find potential taste receptors in flies, we first need to understand how flies taste. Flies taste with their sensilla, which are hairs on the surface of the fly's labellum, as you can see in the picture. The labellum, which is similar to a tongue, is located at the tip of the proboscis of the fly, which is similar to the mouth. These sensilla have specific neurons that are able to receive taste information and detect taste compounds. These neurons are called gustatory receptor neurons, or GRNs.
Fly gustatory receptor neurons are analogous to mammalian taste receptor cells. As pictured at the bottom left, human taste cells receive information through the taste pore and then transmit a signal through the cells to the afferent nerves. Fly neurons receive the signals directly from the food through the tip of the sensilla. Then this same neuron travels up to the fly's taste cortex in the brain, called the SEZ, where the information is then processed.
Our focus is on how we can gain a more complete understanding of the genes that fruit flies use to taste. Specifically, we have a lot more to learn about how flies sense high salts and acids. In order to gain this more complete understanding, we are going through a list of genes expressed in the GRNs to find genes that have transmembrane domains. By using this information, along with the predicted functions and homologies, we are able to identify potential taste receptor genes.
We are specifically looking for proteins with transmembrane domains because all taste receptor proteins are transmembrane. These proteins at the bottom are the taste proteins in humans, or there are some of them, and you can see them crossing through the membrane, which is shown by the green strips. Part of the membrane is hydrophobic. Like oil, it doesn't mix with water. So we can find the transmembrane domains within the protein by finding which parts of the protein are also hydrophobic. Here we have an example of a gene translation. A translation of a gene is the protein sequence that the gene's DNA codes for. The sequence for the protein is made up of many amino acids. The text at the top shows the order of the amino acids in this specific gene, shown by their single letter abbreviations. The bolded letters are the hydrophobic amino acids, and the TMHMM software looks for chains of frequent hydrophobic amino acids, shown highlighted in red. TMHMM also visualizes the likelihood that each region is hydrophobic in the graph below. The thick line at the top summarizes this information and tells us which parts of the protein are likely inside the cell, transmembrane domains, or outside of the cell.
So what Sarah and I did over this summer is we took the set of GRN genes and used Flybase, a database containing fruit fly genes and the genome, to get the predicted functions and translations of each gene. Then we used TMHMM to analyze the translations for transmembrane domains.
This is part of the spreadsheet that lists all of the genes expressed in the fly gustatory receptor neurons. On the left and the right, we have the gene identifications, and it is our job to fill out the columns in the middle. We each got a spreadsheet with about 2,000 rows to fill out this summer. but they are just sections of a larger spreadsheet, which is 18,891 rows long.
When we put the gene identified by the CG number into the database, Flybase, we were able to find information about the gene, including its molecular predicted function, as you can see in the diagram, which can give us an idea of what kind of protein this gene might code for.
Within that Flybase page, we can also find the gene's translation, the protein sequence of the gene.
After getting the translation from Flybase, we insert the translation into TMHMM, which then analyzes the translation to see if the gene has any transmembrane domains. As we stated before, transmembrane domains may be assigned for potential taste receptors. As you can see in the diagram on the left, TMHMM gives the number of transmembrane domains, and on the right, there is a visual representation that shows the transmembrane domains with red segments.
Now, instead of manually filling out all 18,891 rows by hand, I decided to find a faster solution. So with some help from my dad, I'm writing a program to fill out the spreadsheet. The numbers on this diagram mark some major steps of the process that I will go through in the next slide. This platform, called Node-RED, is mostly JavaScript, which is brand new for me. I've only done Scratch and a little bit of GameMaker, so I certainly had to learn a whole lot to be able to do this. What is shown here finds the gene translations to every gene and enters them into an additional content column in the spreadsheet. Each row in the spreadsheet travels through this flow one at a time, and the nodes add or subtract information from it. The salmon-colored nodes contain JavaScript code that I had to write in myself. Here is a visualization of what this program is doing in its main steps. The cells in the spreadsheet pictures are clipped so they fit on the screen, but the text continues. First, the program enters each row and extracts the CG number, which identifies the gene. Then it creates the URL in step 2 and searches Flybase. In the results of the search, it takes out the FBGN number, as seen in step 3, and creates another search, seen in step 4. This search just pulls up api.flybase, which pulls up the protein sequence using /fbpp at the end of this URL, P for protein. So it only pulls up the protein sequence instead of pulling up the entire database page. From this, the program copies the sequence and puts it back into the spreadsheet as seen in step 6. Next, I'm writing another code to enter all of the protein sequences into TMHMM to find out how many transmembrane domains they have. But since TMHMM is not a database like Flybase is, it can't be accessed through Node-RED, which is what I used for this first part. So instead, we are using a Google extension, which can interact with TMHMM and extract the information we need from it. This Google extension is entirely JavaScript, so it is much more difficult. I haven't finished the extension quite yet, but I will continue working on it so my lab doesn't have to get people to do the 300 hours of work that I've estimated filling out this spreadsheet will take.
So here are some examples of the genes that we have found. The first one is CG32853, and its predicted function is G protein-coupled receptor activity, and it has 7 transmembrane domains. In humans, our standard bitter, sweet, and umami taste receptors are specific receptors called G protein-coupled receptors, which usually have 7 transmembrane domains. Thus, due to homologies, this gene here may code for a taste receptor homologous to those in a human. The next one is CG11742, and its predicted function is odorant binding and olfactory receptor activity, and this one has 8 transmembrane domains. In mosquitoes, which have some similarities to flies, there is a receptor, GR3, that is a gustatory receptor neuron, but it's also involved in smell. Thus, this gene here may code for a receptor that is involved with both smell and taste. Here are some more example genes. Both of these genes have transmembrane domains and are predicted to code for taste receptor activity, which makes it very likely that these two genes are involved with the fruit fly's ability to taste. We selected these genes as examples because they have predicted functions related to taste and smell. However, there are many other genes that still have not been tested yet.
Once we figure out which genes code for possible taste receptor proteins, we can obtain fly lines with null mutations in those genes of interest. Then we can use an assay to test to see if fly taste is affected when the encoded proteins are not present. The red gel and the blue gels, seen on the bottom right, are flavored differently, and normal flies show a preference. So if a fly with the newly disabled gene doesn't have a preference anymore, we will know that that gene is required for the fly to taste whatever compound is in the assay.
So when looking at the big picture, we want to know how can increased knowledge of the fruit fly's taste genes assist humans in further research? Well, increased knowledge about the fruit fly's taste genes can help us further understand our own human taste system, because the taste genes we find may have gene homologs in humans. Thus, through this study, we can learn more about human taste preferences, which can benefit companies that make foods, medicines, flavor enhancers, and maskers to appeal to the people. Additionally, a similar creature to the fly is the mosquito. Although flies are not very harmful, mosquitoes are considered the deadliest animal, as they carry many life-threatening diseases, including malaria, West Nile virus, and Zika. Thus, by understanding fly taste better, we can use this knowledge to also learn more about how mosquitoes taste. This way, we can find more ways to mask our own scents and taste in order to protect ourselves from these deadly creatures. Along with mosquitoes, many other animals also use taste to bring harm to humans, such as spiders, ticks, snakes. So a better understanding of fly taste can also lead to a better understanding of how these dangerous animals taste.
We would like to thank John Mack and Yali Zhang from our lab, Paul Breslin, Joel Mainland, and Katie Lung for helping us through MSAP, and my father for assisting me in writing the Node-RED program and the Google extension. This research was supported by individual contributions to the Manel Science Apprenticeship Program. Thank you for your time.
Thank you.